Okay.
Let me know when you. Welcome everyone to this week's edition of Careers in Technology with Al Shpuntoff. This is the Fall Series 2024. I'd like to speak a little bit about Al, who will provide a deep dive into his career as a consultant in bioinformatics, computational biology, high-performance computing, sequencing informatics, including coding and design. He will cover his work as a consultant, contracts with various government organizations, and government projects of special interest, including but not limited to projects to identify remains. For example, he will cover his work with the Henry M. Jackson Foundation for the Advancement of Military Medicine, where he worked with the DPAA project at Offutt as data science and bioinformatics specialist on a project identifying remains of casualties and MIAs from past combats. He will also incorporate his background, how he prepared for this career, and a discussion about technical marketing, exceptional customer support, and first-class technical training and education. I'd like to remind you that this is the fall series of 2024, sponsored by IEEE, IEEE Computer Society, and IEEE ComSoc. Also, funded and founded by Computer and Communications Joint Society Northwest Florida, IEEE Northwest Florida section. These are the complete list of presenters for the fall series. We began with Professor Dr. Victor B. Lawrence. We moved to Professor Paul Berger, Florence Hudson, and William Harding. Now we have Al Shpuntoff, and next week, don't forget, we have Kandahar Nusrat Islam. So please mark your calendars, sign up, and also you can subscribe to the series for your OU. These are the information pieces you would need in order to sign up for each of the individual sessions. And when you want to sign up your OU to subscribe, please get in touch with Michael Viron. This is his email address. Thank you. I'm going to stop sharing now, and I'll turn it over to Al. Welcome, Al.
Thank you. And let's see, are we back to?
You were all set up, Al. You just need to share.
Yes. So I'm going to disappear from the screen for a moment, go back to share, and there we go. And share.
Excellent. We're about to see it, right? It should be coming up soon.
I am screen sharing, yes.
Okay. Al, do you have multiple windows?
Excuse me?
Do you have, oh, okay, there you go.
There we go.
Yeah.
Okay. So, greetings all, and thank you for the invitation to do this. I know that this talk's going to be a bit of a different paradigm from what has been part of the series before. The series has initially been built around a lot of the distinguished visitors from the Computer Society. And I think the majority of folks who presented have had careers in a single institute or in a very coherent path. Mine is a much more adventurous path, and I hope you'll find it interesting and worth spending a few minutes of your time to hear about what happens when you take the other road from the easy one or the one that made sense. There are no miracles in this path. Opportunities appear. I have to grab them. I have to exploit them in order for anything to happen. I make great plans and then God laughs. And we'll talk about a couple of those instances along the way. But every time I thought, well, I know what's going to happen for the next 10 years, something else has taken over. So the background that prepared me for all this adventure, I'm a graduate of the Bronx High School of Science in New York. where I was on the math team. I did a Westinghouse project that involved physiology, treating chick embryos with various drugs and recognizing growth retardation effects. I also did a Ford Future Scientists project in which I wrote a program on the IBM 1620 using a symbolic programming system, in which I made some very elementary explorations in why the halting problem isn't really solvable, but learned that, of course, with heuristics, one could make reasonable guesses at things and such. I wanted to quote my calculus teacher to remind everybody, math is never a spectator sport. It's something that you participate in, you learn. You do not evade when you're doing other things that are mathematical in nature. My early education was an undergraduate during a very political time at the University of Chicago, and then as a math graduate student at the University of Illinois at Urbana. At the time I was at Chicago, the only computer science education was a master's program in information sciences being done for Bell Labs, populated by some very interesting people. And yes, Bell Labs is responsible for a lot of what I learned. So unlike people who at the time studying computer science basically learned Fortran and COBOL and maybe ALGOL, I was exposed to APL, Lisp, Snowball, an early vision of computer science theory. And in my mathematical classes, I got fascinated with algebra, abstraction, and category theory. Joe Goguen was at University of Chicago at the time, and his interests were very much in formal semantics, which was a contagious interest. And somewhere either there or at Illinois, one of my advisors reminded me that category theory did not provide really good opportunities for education. I was made to promise that I'd be happy driving a cab and thinking about category theory in my future life if I wanted to study in that area. I did end up working on category theory as opposed to the semantics. I had an opportunity to spend a year in Zurich and interact with Dana Scott when he was visiting, and with Rudolf Kalman, who was teaching a systems theory class, and with quite a number of other very interesting characters as a graduate student. Nonetheless, when it came to completing a thesis, somebody else created the theorems that I was trying to prove and published them before I got to them. So the work got done, but not by me. I went on to do some undergraduate teaching. The very first task I got was to teach at a small liberal arts college in Sioux City, Iowa, called Morningside, for the two people in this call who know where Morningside is. And I have to admit, this was the most difficult, longest interview I ever did for a job, all for $12,500 a year. The college had gotten a grant that involved bringing a visiting committee in to have them decide whether or not they would start a computer science major. And so innocent grad student coming out to think about a teaching job at a liberal arts college is being grilled by the existing faculty, of course, who want to know that I understand the traditions of a liberal arts college, but also visiting experts from Johns Hopkins and 3 other very distinguished institutions who had very distinct ideas about what a computer science major is. For some reason, after the 3 days of grilling me, they actually hired me to do this. And then they let me see what I had to work with, which was an IBM 1130 and an interested group of students, basically. And so out of that, we implemented within a couple of years the ACM curriculum with the kind of resources we were able to build. We eventually were able to buy a decent computer. In the meantime, there were challenges like nobody wanted to learn the assembly language on the IBM 1130 or computer organization concepts using an ancient machine of that sort. So I settled on, since I was working on this curriculum from ACM, the work by Knuth, which introduced a generic assembly language called Mix, which maybe a few people remember. Anyway, it was great fun. Oh yeah, I needed a computer to run that on. I only had the IBM 1130. So since it had a Fortran compiler, I basically simulated the Mix environment on the 1130. That was the most difficult set of overlays I ever did. I'm glad I'm not doing overlays anymore. Managing memory in the extreme. But I got the thing to run with, as I recall, about 8 bytes left over in memory that weren't in active use. And in the early days of my programming, I think that was one of my proudest achievements. One of the things about running an assembly language class on a simulator like that with a group of students who knew it was brand new was they were intent on finding bugs and proving that the instructor didn't know the machine as well as they did. And so the contest began, and the level of excitement in that assembly language class is something I've never seen since, because they really were enjoying the prospect of showing up their instructor. And they got really excited. Some of them became hackers. 2 of them ran the informatics groups of companies. One of them moved to Alaska and wrote me 20 years later to thank me for getting them excited about computing. So some magic happened in that classroom. And I think that the lesson I took away from that was you have to inspire people who you're teaching to become excited about the subject. It's not about presenting just material. It's about exciting people and turning them on. And that turned out to be important later in my life as well. I moved on from Morningside down 90 miles to Omaha, where I am now, but it's been a long time and many places before I came back to Omaha. At the University of Nebraska at Omaha, I ended up inheriting, once again, the assembly language and computer organization class, but this time we were at least using IBM 370 assembler, though on a machine at Lincoln with a remote job entry. One of the things that I used this course to do was to challenge students who had previously done all their exercises and never had to do anything besides keep it in their head and write some clever code to solve simple problems. So the assembly language class became a place where they had to do a project that was large enough they couldn't keep in their heads, complex enough that they had to understand about writing individual parts of the code and tying them together. Oh, and yes, it had a hook. The name of the program was Robots. It was a simulation of a robot war. And I have to admit, 15 years later, I walked into IMSL to do a training class for one of the computer companies I worked for. Somebody from the back of the room saw me walk in and said, "Robots!" to remind me of the code that they'd struggled with. a lot of people have written back to say, yeah, they hated the program at the time, but in future life, it was what prepared them to write large code and be part of larger systems. So again, it engaged people, not necessarily in their enjoyment, as other things have proved to be. One of the themes that enters into my life about this time is that of beginning to teach people about other programming paradigms besides the standard procedural languages. As I said, I was early exposed to functional programming and Snowball, which was a language based on pattern matching and artificial intelligence type logic. Every statement had a potential pattern match between strings and a path to follow if there was a successful match or not a successful match. And those matches could be recursive, leading to unfollowable programs, but delightful fun to try and construct the path through a program. I had the chance to teach from the Abelson Sussman book on Scheme in a year at Oberlin, to teach functional programming as a first class and integrate semantics into teaching programming languages. So a great variety of things, which certainly prepared me for a bunch of what was to follow. I guess this is a good point to ask if there's discussion to follow. All right, so up till this point, I'm pretty sure I'm going to make a career of teaching undergraduates and graduate maybe. And then I met somebody, got married, and she wanted to go to grad school. She ended up getting into Stanford and an MBA program. So I needed a job in Silicon Valley or somewhere nearby. And Ardent at that point was a very promising startup, the startup that couldn't fail. Surely everybody that's been involved with a startup has been involved with something that couldn't fail and did. But anyway, it had blue ribbon credentials. It was funded in part with Japanese money from Kubota. The principal architect was renowned for his involvement with digital equipment company formerly. The vector unit was designed very carefully. They used the MIPS chip. And so it was a small company with a very large reach to try and create affordable supercomputing. So it was modeled on the Cray, basically what it could perform well with were those things that could be vectorized. Randy Allen was hired as the compiler architect, and his first vectorizing Fortran compiler was done for Ardent. My role was to be the education, training, and post-sale support guy in terms of making it programmable, explaining it, and helping people to gain an understanding of how to use a rather sophisticated instrument. Alan Michaels was the founder of the company and a bombastic guy from the Bronx, as I recall, who had done a number of startups. One of his marks for this company was, oh, buy it. You have 30 days. You can turn it back if you don't like it, if it's not performing for you. Well, you can see where that's going. When somebody would call after 30 days and say, this really isn't working for us, the next question was, can we send Al and have him work with you for a little bit? And so without much warning, I would be in a new environment, talking some new kind of science that I probably had a very vague background in, looking at some code that inherently wasn't being vectorized, and running a profiler to understand where was this code spending all its time. And so the question was, in a week of doing a few lectures and training people and looking at critical pieces of the code, could I get them excited enough that they would keep the machine and continue trying to make use of it? And I saved about 29 of them before Ardent had merged with another company, and its vice president made promises that nobody could have kept. In any event, it helped me to understand many of the issues in optimization and many of the things that later got addressed in architecture, but which were very much evident in the early efforts in trying to make vectorized computing something that worked for the masses. In the end, it wasn't our marketing that failed. It was somebody else's vision of trying to take the same space. I'm sorry, somebody said something. No, okay. Anyway, it was somebody else's vision, namely Silicon Graphics, of making the same space more accessible without the need to worry about vectorization. And so it was also built with the MIPS chip that ran our machine, but it had graphics units as opposed to the sorts of, as opposed to trying to do graphics processing on vector chips. Nonetheless, visualization ended up playing a very important role at the company. I liked our T-shirt that we produced in extra large size. We have the balls for supercomputing because we could do reflectivity of the pile of silver balls display really well. And the first time we went to SIGGRAPH, that was largely our demo, was how we did ray tracing and could actively present good graphics. The second time, we thought we would basically show off what we could do with 3D graphics. And so we created a core of marketing demos that did things like slicing, dicing, putting up multiple dimensional data in a form where you could take different visions through it. And we all tied it together with a little bit of a data flow where we could say, well, first you put a filter through it, and then you do this, and then you do that, and then you display it, and then you have these controls to move it around. AVS was a great hit, and in fact, in the end, outlived the computers it was designed to demo. It continued through the time that Stardent and Kubota became the inheritors of the Ardent intellectual property. So among my tasks, once again, was to get people excited about doing something they hadn't done before, understand from mathematical foundations that multiple dimensions meant interesting geometry, and that there were good, interesting ways to display things. So this was an early example of lots of good things: visualization, data flow driven software, and display tools, all in an affordable package. The next startup I joined was MassPar, massively parallel machine built on a SIMD model. We used to compare it to the Cray as The larger supercomputers were large animals, and our machine was like killer bees. A flock of killer bees could be just as effective in taking down a rhinoceros as the rhinoceros was at charging ahead. So it was a single instruction stream with parallel execution on different data streams. To build this market, once again, the challenge was not to be academic about it, but to cast this programming model as both a way to think and something that could be practical. So we needed to have examples in which SIMD thinking actually made sense as well as performing well. And this was a challenge to create course materials and exercises that forced people to think in a parallel way. When I first joined the company, they were on the other track. They were trying to sell it to academics by looking at the technical aspects of things. And that didn't fly very well with much of anybody. Bored people to tears, did not get people excited, and didn't show people the performance that was available on this machine. So the tools to debug, to understand, had to feel natural. And again, that meant training people in how to use them and showing how they were used for the purpose of actually solving problems. Ultimately, though, the future of the machine ended up depending on finding some niche markets where this kind of architecture actually spoke to the problems. And so in particular, we brought a couple of people aboard who developed golden eyes for doing video compression. And we were producing the highest quality video compression around. And if only 2 Christmases sooner, people had started buying video discs, MassPar might have been the telarc for the future. That didn't happen. There was a group that was doing a data warehouse and demonstrating that they could produce joins and other activities needed in a data warehouse faster than Oracle could possibly do them because they had so many engines doing these things. Again, there were others who did it better, so that didn't process really well. Signal processing was what they were going to raise the next round of capital based on. It was incorporated in some flyable systems and in earlier versions, or the earliest versions of the systems that shot down missiles in flight headed towards you, but eventually was replaced in the Iron Dome and other things of that sort. Still, it was clear that the future of MassPar was going to depend on finding markets and exploiting them. What better way to do that than to have your customers do the initial research for you? That's right, we held a contest over the summer for students who were at universities that had one of these MassPars, and we promised a free machine and support to whoever gave us the best application to develop going forward. And we had a winner in the contest. The winner was genomics, and in particular, the idea of doing homology search. Now, for those that are not working in bioinformatics, this originates in a number of ways, but we have to take ourselves back to the time when sequencing the genomics of E. coli was a PhD process. You got an entire degree from sequencing a couple of genes in E. coli. It was that much work. We knew it was a double helix. We knew it needed to be flattened. We would get a sequence of what was there. And then somebody else would do a project. They would start with sequence, they'd flatten it, mostly do it right. They'd get a sequence. Question, are the two sequences the same? Unix people knew the answer. You used grep, right? Very easy. Except then the biologists started talking about life. Well, sometimes there's a little gap in here and we need to still be able to recognize that it's the same sequence. Sometimes we get the loops wrong. And oh yes, there's two complementary versions. There's this one and the complement on the other strand. And sometimes we get them so that we want to know they're the same, even though we sequence the other strand. So what we got instead of a grep kind of problem was grep with changes, things missing, and so fairly soon the idea came forward that what you're really trying to do is align the sequences, not compare them for equality. And so if you could find islands of alignment, gaps, and then more alignment, you could find what was dubbed homology. The initial problem was posed to Michael Waterman, who at the time was teaching statistics in North Dakota and was on sabbatical visiting Boston, where Temple Smith posed the problem to him, and he applied an algorithm of dynamic programming, which he had used in other fields. Well, the algorithm worked ideally well. Software was developed to do it. The program FASTA by Bill Pearson from Virginia was created, and dynamic programming came to be the best possible way to align two sequences, and it became the standard for genomic search. Well, where there's an exact answer with bad complexity characteristics, there's always a heuristic answer as well. And a government agency developed something called BLAST, which was based on heuristics rather than doing the full dynamic programming. The version of what we received was the full dynamic programming. And since it was too slow on standard machines, what we were able to do was to do significant parts of it in parallel and search the entire database, doing the necessary filling out the dynamic programming matrix with relative speed. The result of this is that we sold about 45 of our machines into various institutions that were doing genomic research and that were doing parts of the Human Genome Project. And that needed huge amounts of support. I mentioned that at the bottom here that this gave me an appreciation for careful data curation, because the data was being created by whatever means could be utilized. Eventually it was automated some, but I do remember a lunch in which the director of the Swiss Bioinformatics Institute spent about half an hour of the lunch complaining about the lack of quality control in French robots that were filling the database with junk genomic sequence that was not well annotated at all. And so matching against that proved to be a real waste of time for people doing genomic work. So the curation became an interesting problem in supporting the machinery. Never have too narrow an approach to supporting something. And indeed, one of those midnight phone calls arrived one night from my marketing guy, who's now a distinguished professor, I should admit to, in Tokyo, who was saying, our machine doesn't seem to work when it's searched with this against this database. Find out what's wrong and tell us by 8 in the morning your time. So I spent the whole night running my searches, discovered something had been put in the database with an empty header, and this was breaking the algorithm because the code was not robust enough to recognize a missing header at that point, and was falling apart. So data was important. Where genome annotation came from became a matter of interest. And looking at various versions of the algorithm, which allowed us to do greater discovery, also became one of the passions. We ended up selling machines to the US Patent Office, which were used to help understand when a sequenced gene was actually unique intellectual property as opposed to a normal thing that one might find by following one's nose. And so determining whether there was unique art to genes ended up being part of the search process. The base algorithm of dynamic programming also proved to be important in some other areas, notably speech recognition, and the technique known as hidden Markov models used a core of a very similar algorithm. It was adapted by folks at UC Santa Cruz and elsewhere to help to define protein families and to recognize weaker members of protein families. So rather quickly, the genomic search also became something involved in the analysis of proteins. Well, that looked like it was going to be life going forward as an independent, as an employee of MassPar, except they went to the closing for the next round of funding, and a defense contractor who had incorporated the MassPar machine in their design pulled out, saying they were never minority investors in anything. And so they weren't going to invest in this. And so the whole of the next generation of MassPar's hardware went poof on I think a Wednesday. So by Friday, the engineers were being laid off. The various people were trying to reorganize. People who'd done really interesting software were organizing a successor software company to do early decision science. And then there was a question of what do you do with me? I was doing implementation projects under contract. MassPar did the contracts, charging $150 an hour for my time and paying me $60. Sort of typical markup by various folks that do the business end of things. So they made me an offer I basically couldn't refuse. They said, we'll give you the contract if you can be a business by Monday. And so, yes, indeed, I visited the Santa Clara County offices. And by Monday, I was a business, AFS Informatics. And I picked up the contracts. And, this was sort of viewed as being, yeah, you'll keep everybody going for 2 or 3 years. You've got about 3 months of contracts ahead of you. And so you should be able to make something of this for a little bit. Well, 10 years later, I was still doing it. The actual path was to develop some genomic tools to do migrations from this machine to the next fastest version at TimeLogic, to also get requests for solving other problems from the customers who'd gotten to know me as their MassPar guy. And so, despite all the things they tell you in business school, after all, I had a wife who'd gone to business school, do all your planning, no. I was a business between Thursday and Monday and had to learn how to do it all myself. Manage contracts, do my own business development, get the appropriate insurance, especially health and liability coverage, do my own collections, and make critical decisions about what did the future of a business bring. This is a day when most of the genomics community was still a very small group. So one of my early stories as an independent was buying a t-shirt at the local computer store with Ratbert sitting at a large desk with a tablet or a computer on the desk and nothing else. And basically saying, I'm a consultant. And I wore it on the show floor at the Genome Sequencing and Analysis Conference. And by gosh, people would stop me and ask, are you really a consultant? Could you help me with something? Or I would start a conversation with, so what are your points of pain? How could I help you? And out of those conversations, I'd get 3 or 4 contracts and keep myself busy for quite a while doing really interesting work. Expanding beyond simply the hardware to build tools around the hardware and to help to solve real problems that people brought me. The downside of being an independent consultant versus a developer of software in a university, well, of course, there's no security in it other than continuing to be able to sell yourself. And you don't get credit for a lot of what you do because people buy your software, incorporate it, and publish the research. And the software that helped them was just something that they used along the way. You don't credit the typewriter manufacturer. So a lot of this was sort of open, and it was useful to be able to walk around the show floor and people would say, okay, maybe you could help us. Is there anybody here who can vouch for you? And you'd point at 3 other people on the show floor who you've done work for. And yeah, those are good references. Yes, yes. All right, so what goes right, what goes wrong? TimeLogic bought 150% of my time, and I forgot about doing business development. And then TimeLogic dropped me, and I'd burned my network. I worked for a pharmaceutical company under the authorization of their vice president, who left to go work for a competitor. So all her contracts were canceled. It took 6 months to collect her work done for a variety of things. Yeah, collections is sort of automatic, right? Not when you're independent and not when interesting things start to happen. Unforeseen disasters. A company that we worked with was just trying to go public. No, actually, they were going for their final round of seed money, and they'd successfully raised the next round of seed money based on the work we were doing. And so they had bought add-on software, several licenses, everything was looking rosy until they went to close the deal at their funders in the World Trade Center on September 11th. And so, in fact, that company never recovered. There was nobody to sue. And I lost about 6 months worth of work as a result of unforeseen disaster. Large company maybe has insurance against that kind of thing. I didn't know to do that. And so that was hard to recover from. All right. I mentioned MassPAR's paradigm. The next implementations of Smith-Waterman and HMM ended up being on field programmable gate arrays, and our competitor used application-specific integrated circuits to provide essentially the fundamental operations in hardware. My contribution here, including demanding that various bits and pieces of the algorithm be done this way so that we could create discovery environments, was to create an interface so that it could be called and integrated into HPC environments. And I built a number of tools based on the basic engines to help with the discovery mechanism. I'm going to jump a bit ahead now because I know David, if among others, wanted me to talk a bit about this part. Suffice it to say that we actually, that while I was with TimeLogic doing the FPGA-based product, one of my favorite moments was a blind demo where I challenged a group at a pharmaceutical company. Okay, you're probably working on some compound or something. Give me a sequence that you're interested in searching to find out what it does. And I'm going to show you an algorithm you've not seen before. And well, yes, they gave me a sequence. I put it in through the algorithm. And while I'm doing my little song and dance about, so this is what we've done. We've looked at all of the SwissProt database, and here are the things that are potentially coded in your genomic sequence. And the fellow who'd given me the sequence stood up, said, "Al, you're fascinating, but I have to go to the lab now," and with emphasis, and he walked out. I asked him like 5 years later, so did anything ever come of that? And by then it was an investigational new drug that was being taken forward. I wish I could say it was a successful investigational new drug, but in the end, it had some side effects. And so it didn't cure a disease that I have. But nonetheless, it was an exciting moment to discover the potential of something that was being investigated in the lab in real time as part of a demo for what we were trying to excite people about. And I can think of no better definition of exciting the audience than giving them a lead to follow as a drug company. Okay, deep breath. Questions?
So tell us about the salmonella contamination. I really want to know about it.
I'm sorry?
Can you tell
No.
us about the salmonella contamination?
Okay, so in this phase of my life, I went to work as a federal contractor supporting the FDA's Center for Food Safety and Applied Nutrition. And they were doing genomics work. One of our major tasks was to understand salmonella contamination. And so one area that we were able to get lots of samples was salmonella in the wild. As we cataloged salmonella contamination, we would sequence each of them and run them through a pipeline that identified what are called SNPs. That stands for single nucleotide polymorphism. That is the simplest kind of variation in genes, where in the long sequence of bases that are present in a gene, one of the nucleotides changes, and then that changed nucleotide is inherited down to the progeny of that particular organism. So SNPs turn out to be relatively useful in identifying the heritage of different groups further down in the chain or in the genomic tree. We made the SIFSAN SNP pipeline available so that agriculture departments could run their sequences when they found things, and they would also contribute to the database. Now, the advantages of what we had, using genomic search, we were able to very rapidly identify what were the closest cousins to the sequence that was being examined. So in the example that I've told David about in an earlier discussion, the report was made of what appeared to be salmonella poisoning coming from candy apples reported in a safe way in northern Minnesota. And so we got the genomic sequence done pretty quickly and looked for where's the closest cousin. And so the next thing we discovered in the database was, hey, yeah, we've got something that looks pretty close to it. And where was it? Well, it was in San Diego, California. And so the next question went to Safeway. Does something in your candy apples have to do with a warehouse in San Diego? And within a day, they not only identified what was the source of their salmonella problem, but they'd managed to recall all the appropriate candy apples and went forward in health.
That's fantastic. Can you speak to prior to the recent global emergency? Remember the global emergency we had for a while where there was a COVID-19 that was sort of shutting down the world? Before that, there was a lot of discussion about things like C. diff, for example. Do you think that being able to track C. diff might be able to help people to find the sources and the spread of C. diff and help hospitals and nursing homes and other places to curtail these problems?
Well, the transmission medium for these pathogens in food is fairly limited and much more traceable than are the ways in which flu and the like spread. So it's much harder to track the specific organisms and to have the full and full sequences still turned out to be very helpful, both in terms of finding something to counter them and in terms of understanding when there were new sequences showing up and new variants of flu. So all of that process is mimicked by other agencies. In the food safety world, bureaucracy gets in the way. Initial reports go to the Center for Disease Control. For example, right now there is a foodborne emergency having to do with the beef or the onions or something of that form in quarter pounders being sold by McDonald's in the Midwest. E.
And
coli is
also my favorite food, waffles.
and actually that's a salmonella issue. And yes, that is something that will be tracked by the FDA center and the software I've been describing. The E. coli, on the other hand, goes to the Department of Agriculture, and it's a different group of people who are charged with recognizing how that bit of contamination ended up in the food chain. So
So do these agencies work well together?
the agencies do work well together. When CDC first has an inkling that something is going on, they notify the appropriate agencies to be ready for what was going. And in fact, we conducted one of our tests of Amazon Web Services with the idea, suppose there's a widespread food emergency and we need to do all this processing in a weekend. If we needed to turn to AWS, would they be equal to the task? And we could prove that yes, they were able to process as much of the data and assembly and make the trees as was necessary to deal with what might be a countrywide or worldwide emergency that it showed up rapidly.
How does that feel to you to see where you began in your work and how AWS can provide such resources, ad hoc? How does that feel?
There have been a number of times throughout the career where I have kind of looked back at things that were a tremendous amount of effort at one time that just happen instantly now. I mean, I think about, for example, working with AVS and what a struggle people had wrapping their heads around some of the 3D visualizations. And now it's just old hat. It's on every Mac and every PC as an available tool with your choice of a dozen kinds of software to produce variations on the visualization. So people don't recognize just what a significant effort it was to incorporate the mathematics, to figure out how to render things, how to present them, what kinds of geometry actually spoke to the data and elucidated what you were doing. And now people just look at the display and go, yeah, that makes sense. So yes, there is an awful lot of times that you kind of look back and go, we've built all of this along the way and people will never understand what was involved in so much of it. That's part of why I mentioned things like putting together the overlays in my program back at Morningside. Yes, we still have to occasionally rethink how we organize memory, but it's not at that granular level that it was way back when.
But also the conceptualization of the problem you're solving. You swim in that ocean, and you've been doing that for years, and many people may not really even have an understanding of that problem domain. So using the tools isn't the same, right? Using the tools isn't the same as understanding the ocean.
That's true, but there's a flip side to that, which is people who have been trained from the beginning in the exact problems that are being solved now and have as their foundation things which we fought for, but now from that foundation, using those tools, are able to contemplate much more sophisticated problems. So
Can we move on to the DPAA and identifying combat?
okay, I'll try not to take too much longer, but yes. So one of the most recent projects that I worked on was with the Department of Defense, accounting agency for POWs and MIAs. I do have to apologize that I'm going to be more vague in this section. I'll give you a couple of references at the end that allow you to dig down a little bit deeper. I'm going to try to introduce the problem and regrettably not be able to tell you much about the solution for various reasons. All right, so the task of this agency is to go back to sites where there were MIAs or POWs lost as early as World War II and up through the present day, but mostly ending with Vietnam. These remains have been the opposite of carefully curated. Many have been mistreated, some have been out in the wild, some have been in locations where dead animals ended up in the same burial. Some have ended up with various mistreatment with chemicals and environmental factors. But we find locations like in German forests where there are the remains of an Air Force bomber from World War II. We're still excavating parts of Pearl Harbor where large numbers of people died in the same location and their remains were commingled. A recent find was to raise from the bottom of the ocean a Japanese prison ship, which had some 300 Americans imprisoned on it and was bombed at sea without anybody knowing that there were prisoners aboard. And in all these cases, the task is to try to identify the remains. When you succeed, sometimes they're as significant as Medal of Honor winners. Sometimes they're simply people who were lost and their families have always wondered what happened to them. It was a very satisfying and important task to perform for the military and all those in their families. So providing long overdue honors and helping to ensure that some of the unknown didn't remain unknown. So this was a regulated forensic laboratory with important contributions from many different kinds of professionals. We start with the historians who were able to take military records and do reconstructions of the occasion on which people were lost, or provide at least lists of people who were potentially the victims that we were trying to identify. And we have forensic archaeologists whose tasks were to understand the sites where we went to retrieve things, and anthropologists who were trained in recognizing and identifying remains. There were dentists specifically to identify unique characters of teeth. There were histologists who were capable of taking small samples of bones and determining such questions as what was the origin of the particular victim, what was their typical diet? Did they grow up eating mostly rice? Did they grow up eating mostly grain in the style of Americans? Did they, the chemical composition of the bones is determined by what you ate while you were growing up during the periods of rapid growth of bones. And there were bioinformaticians and data scientists, which is how I got involved in something this unique and different. There's a very careful process that had to meet the standards of a forensic laboratory. That is, if you were to have a criminal trial about something, would the standards of identification meet what would be required for a criminal trial? Which meant that some of the techniques you might use in a less formal setting are not sufficient when dealing with human remains in any sense. The DNA processing, basically, this lab was responsible for extracting parts of the bone that hadn't been completely rotted, and then the samples were sent to the Air Force DNA laboratory, and that's in Dover, Delaware. They have a lovely web page that I've referenced here, which gives you basically an opportunity to walk through how they process the DNA, what the improvements in that process have been, starting or around the mid-2000s, where they were able to actually retrieve useful DNA, both from the mitochondrial and nucleic parts of the bones, by more sophisticated techniques than had been used before. DNA is not all that useful unless you have references to match against. So it took the cooperation of surviving relatives, and then based on whether one was looking at maternal relatives who offered samples or paternal relatives, there were different tests that would be run to show if there was a properly inherited trait or sequence from that particular parent. For more recent conflicts, part of the military processing is to take blood samples. And so there are good samples for more recent MIAs or more recent casualties that are identified. But if you go back to the records from World War II, you have mass inductions of many people. Similarly, during the drafts for Korea and Vietnam, the records are a little more challenging to make use of as you try to do the matching. But with the historians and the physical evidence that's present, lots of things become possible. So just to highlight the area that I tried to get involved with, how does data science get involved in all of this? How does bioinformatics get involved in all of this? The historical documents typically get digitized, and then making them most useful involves enhancing the quality and doing many of the machine recognition techniques, which are useful in dealing with documents. The informatics requirements are extremely difficult. Physical evidence is of many varying kinds. The samples that you do acquire need to be very carefully handled, because after all, you're trying to assemble the proper collections of remains to be buried properly once identified. And so accounting errors are not permitted. One must be very careful in the tracking, the matching of measurements and analytics, many of which are done at remote sites and then brought back to all of the items in the inventories. There are multiple kinds of potential analyses. No sample having all of the analyses done. And one ends up with an assembly of evidence towards identification, which in the end has to be approved by the medical examiner based on all of the available factors and what they tell you about it. So when you propose to do machine learning, the standards that you're going to try to meet are more stringent than in trying to prove a ROC curve is suitable or that something is interesting. You have to prove validity in statistical senses, which tends to be a relatively difficult task.
Is this one of those cases when the alignment helps you, like when you were talking before about aligning things in the DNA?
Indeed, when processing the DNA, you have to have more knowledge about the DNA than simply, oh, here's a sequence and here's a sequence in the database. What you need to know is relative frequency of particular kinds of variation. And you have to have some sense of why is this one of the things that is preserved as generations go forward, so that what was present from the mother of the victim is also present in the other progeny of that mother, or what was present in the father is also present in the progeny of the father, and is something that is largely preserved through several generations. So the knowledge of genomics is much greater than simply the original knowledge of is there something resembling a match. Alignment is still key, but the changes are things you have to recognize a gene spans across those gaps, and you have to recognize that a gene continues even though there's a variation in some places. Over the years, we've come to identify much more variability in how genes are used, so that we've discovered, for example, that there are other things that help control how the coding occurs. And so the genes that are actually active and hence preserved in replication are more interesting than simply, oh, here's a gene in the database, but rather here are things known to be preserved and useful in the identification process. So it's gotten much more sophisticated in the time since we were first trying to identify what sort of sequence was there and what kinds of functionality were there in the genes that we were identifying. Okay, so did that answer the question, I hope?
Yes, and like later on when you finish your talk, if we could get some references so we could learn more, it would be great, if you have anything like that. Thank you. Thanks.
So anyway, a sample issue for machine learning. Over the years, and this has like 100 years of history behind it, criminal labs have always tried to identify the body by having an idea how tall was the person before they died. And so what can you, when you can't assemble the entire skeleton, the question is, what can you predict from the size of some of the longer bones, the larger bones, to estimate the height so as to match against records like the enlistment or the medical records? Because of the ways in which it's done, each small community, each region would create its own traditional charts, formulas, most of which involved linear regression. So in Thailand, they would have a national database to be able to predict for Thai backgrounds what was the relationship between the length of the tibia and the overall height that you could expect. These are all predictions, and the predictions that were based on different racial assumptions for the same measurements significantly different from each other. So the question arose despite the 100 years of history in which this was used as a basis, and recognizing the changes in culture and biology, surely there must be a better way to identify natural groupings that has more validity than the traditional use of race and ethnicity. People who are of Chinese origin but raised in the United States, like McDonald's, like to eat Western diets as well as traditional Oriental diets. People who grow up in the United States of traditional Eastern European backgrounds may develop a fondness for Japanese food and sushi and the like, and may turn out to eat a diet that isn't as typical of the traditional people in our culture. So the question is, if you let the data speak rather than letting old history speak, is there a way to identify natural groupings that has greater validity than race? Is there something inherent that will emerge as a clustering basis? Does the data have natural clusters in it? And is there a better way than simply linear regression for identifying the factors that in fact will lead to better predictions? Is there some way to incorporate, whether it be the histology or other factors, to produce better estimates? So the opportunity for data science in contributing to this area is strong, but so are the judgments for how do you recognize if you've succeeded or not. And I guess I'll stop there because telling you more about the inside of that organization probably violates the security clearance. So.
Thank you so much. I know I am very intrigued by this entire topic, every little aspect of it, every single aspect of it. And I'm quite sure, since we have retained our audience, there are many people on this call who are also quite intrigued. Would it be okay if we stop sharing so we can see the people and bring everybody's face online and ask questions? Would that be okay?
That's fine by me.
Excellent.
So.
Can you get excellent? Hi. So, Li Feng, I know you have an interest in this area. Do you have a question for our speaker?
Yes. So thank you for this great talk. And Al, I want to know that since my research is also related to the bioinformatics and AI related stuffs, so I want to know what's your vision about the AI for the bioinformatics or the molecular AI?
Oh, difficult topic because I see less utility in large language models than I do in some of the other areas that have been recent examples of wonderful tools. For many years, I was involved in a competition for the assessment of protein annotation, the contest known as CASP, where everybody used the techniques of the day to try to recognize both the folding that proteins would take and the function that was implied by that and the ability to interact with them. AlphaFold is a revolution. It has provided a new level of performance way beyond anything that we were able to achieve with our ad hoc techniques, with hidden Markov models for families, with recognition of some of the statistical tools that we had before. So there are some spectacular examples like AlphaFold of successes for artificial intelligence in bioinformatics. Imaging is another area where the fine eye of the best practitioners is still capable of finding subtle things that AI does not. But on the other hand, AI is well trained in finding abnormalities that might escape the human eye. So that's an area where tremendous progress has been made. And for having even those two, we can say AI has been very successful in bioinformatics, I think.
I see. Thank you so much for this. You know that especially the triple AI fellow, like the Jeffrey Hinton and also the John Offutt, that this year they received the Nobel Prize in physics. So this is actually very different from usually that people thought that this is the award is given the mathematics or the physics peoples, right? So this changed the path of the AI, also the people's impression of the AI. That's why recently I found that a lot of people, especially in the medical field and other fields, that they started to also trust the AI, although that this is just the initial step, but this really changed the people's mind and thinking thoughts. And especially I feel that like the 10 years ago or the 15 years ago, when I worked in this is the medical field, that the data is very limited and it's very small. For example, that we just work on the 13 samples of the patients to do the disease prediction or something. Right now, it's kind of like that the AI model, especially that they requires a lot of data, right, to do the training or something like this stuff. So, but I feel it's still very difficult or challenging to collect a very large of the data if we just worked by ourselves or very within small communities. So do you have any suggestions about that? How should we I know that we should very actively work with other people or explore the opportunities from different institutions or see the collaborations opportunities within them. But do you have some specific suggestions or insights about that? So how should we expand the data? Since even that recently some people try to build the biomedical database or something to try to fit this trend. So do you have any suggestion for this path?
Oh, the thing I recommend to people probably most often is to try to get involved in the activities of more of the professional societies that support these activities. So there's an international society for computational biology, which every year has a conference called ISMB, Intelligent Systems in Molecular Biology. And it's organized around special interest groups, including specific ones having to do with understanding gene function, having to do with machine learning in bioinformatics, and other such areas. And so the best way to really understand how many different directions all of us are heading as we try to exploit artificial intelligence in biology is to interact with a lively community like ISMB. I always had dreams that EMBS would be more of that kind of group. And indeed, in the early 2000s, the Marsteins and I and others tried to create an IEEE society on bioinformatics, but the conflicts in politics turned out to be just too much.
What's nice about it is that there are data port platforms that if we were able to bootstrap some projects, we might be able to create databases that could be shared and tools that could be shared and some tutorials by Al and other people who can help us to understand what we're doing. you know?
Indeed. One of the difficulties of doing databases, though, that has really come to bite this community is that when you begin the process of curating a genomic database, you're making a commitment for a long period of the future, and so are your funders. And so we've lost many of the funders that were responsible for keeping things going during the 1990s and the 2000 to 2015 era, so that we're looking for new sponsors. We're looking for people who actually understand why these databases are important and why keeping them up to date and making them current is so important.
And not just for animal models, but also plant models and other models that might be just as important, that may have more sources of funding, for example. You
Well,
know,
even what was once the well-funded, or where somebody's made a huge investment in sequencing a particular organism. I was personally bitten by doing an awful lot of work for Novartis, which became Syngenta, sequencing rice. They obtained the full sequence and then locked it away in the vault after publishing in Science. And the hope was that that would lead to all sorts of advantages in the world, but instead we ended up with another product in the Syngenta vault.
I'm
Well,
sorry.
China bought Syngenta and now owns the vault, but
I'm sorry.
Thank you
Is
so
there
much.
anyone else who has a question, an interest in this field?
Probably not in the field, Kit,
Mhm.
but Al's definitely created a lot of thoughts, and it's really intriguing to hear. I have a few questions, but that may not be directly in his field.
Okay.
A
So why
couple
don't we
of
hold
them,
them? Why don't we hold them until we have covered the things that are in his field, and then we can come
okay.
back to this one.
Well, he's definitely handled them, so they are more soft skills than hardcore bioinformatics skills.
Right. And I just wanted to ask Dr. Kelwani for his point of view in terms of the history that's involved. Do you have a observation or a comment that can help us see your point of view about the history that Al has covered today? Are you there? He might be quiet. Sorry. I might have caught him off guard by asking a question, a directed question. Anyone else who has an opinion about the history that Al has covered? So, I mean, there are many aspects of history: history of the technology, history of the progress of these jobs, history of the changing environment, right? But also the history of those people who went missing, right? And how we can help, like, part of what you're uncovering will change the way we understand history of the things that happened there, probably, I would think.
it does indeed. The discovery of artifacts, it's important not to lose the past, and it's important to celebrate the heroes of the past, and not simply declare the missing. So,
Yeah, my uncle, I was mentioning in the chat that my uncle went down in Typhoon Cobra in the Pacific with a lot of other people. And they apparently were sent out after down ballasting to refuel into the storm, and known they were known they were going to die. They knew this. Everyone, they were sent to die, so information would not be passed to the enemy. And no one has recovered those people. And it's very,
it doesn't mean they've stopped looking. And that was, I think, one of the, as we are able to search in new ways, as we're able to find DNA in samples that have been mistreated in different ways. I mean, right now, the Japanese prison ship is actually demonstrating techniques which can be exploited in the next finds to deal with bodies that have been underwater for a long period of time and other such challenges.
and you were talking before about like limited records, lots of mass enlistments, and the military's incredible ability to lose track of things.
I should say my personal discovery when I took this on was the completeness of the records that they did have. They were able to bring forth the nature of battles that had occurred 50, 60, 70 years ago, and to describe how people were lost, to know where the missions were that bombers were intending to go before they were shot down, to understand what role they were playing. And the historians were experts at combing the military records to find much more of the story than I would ever imagined existed.
That's wonderful.
So it is a job for a professional historian with the tools, techniques, and abilities of somebody really dedicated to military history. The anthropologists involved in this have built careers there to understand what they're looking at, what techniques will be helpful in identifying remains. And I'm in awe of almost all of them. My own background, I was the kid of a dentist. And so I was really interested in a lot of the forensic dentistry issues, which were absolutely fascinating. We had some old textbooks in my library, and I brought them in for people to look at to see, here's the techniques that were used in doing dental fillings in the 1920s and 1930s. And why is that relevant? Well, you find a unique dental filling in somebody's mouth, and now you can connect that to standard dental practice in that period of time.
Yeah, I mean, you're reminding me about many of the sites around Europe that I've visited that many people here on this call may have seen, where you have these ossuaries and like where people were held before being destroyed in camps in Europe, for example. And there are parts of their body there that were preserved, like teeth, there in piles, and other belongings that probably have DNA on them that,
Not at this time. It's unlikely that possessions show DNA at this point. One of the confounding things about the way DPAA has to deal with things is just because you found a tooth and a bone in the same grave doesn't mean they're from the same person. You
so
actually,
things like the soaps that have been made from people who are prisoners, would there be DNA in that at all?
I don't know. I'm sorry. That's the rendering processes typically destroy DNA, but some things, one of the things the Air Force lab points out is that sometimes the mitochondrial DNA will survive where the nuclear DNA did not. And that may help if you have a maternal sample to compare against.
Thank you. Thank you. Are there any other questions? I just want to congratulate, thank you for what you have done. Congratulate you for your achievements and thank you for what you've achieved.
Well, I thank everybody for listening and for finding at least part of what I did interesting here. It's always strange when you're getting older to know where the differences between telling interesting stories and meandering out in the ancient past in things nobody cares about anymore. So I thank you all for listening and for your interactions.
Anyone else have a question? Yes, Stan. You're on mute. You're on mute, Stan. You have to unmute. It's not coming. Oh, there you are. Okay.
I'll also release my brother from his vow of silence. I know he's on the call. If you'd like to ask anything, I'd be glad to answer something from
I'm sorry,
somebody.
Stan, your microphone doesn't seem to be connecting to us. I apologize.
Is he asking his question in chat, maybe?
No, Stan is, his image is up in this part of my screen. I don't know if you can see my cursor moving here. No. Stan is in the, Stan, would you like me to send an email to you with Al's contact information? Would that be helpful? I'm sorry, I can't seem to fix this problem. I don't think he's muted.
Let's thank everybody. You've stayed well past the advertised time, and you've
And
made.
your brother is here. Is your brother here? Does he want to say something?
Is he still here? I'm not sure.
I can't tell.
No, I think he disappeared finally.
Yeah, I'm still here.
Oh, hi, Walter.
Hello.
Hi.
Nice to meet you.
It's been same here. It's been kind of cool to listen to a lot of the details that I haven't heard before. It's quite a history having lived through it and his challenges, but I guess the takeaway for everyone is that sometimes things you work on that you feel are important may go unrecognized, even though it turns out that they are.
That they are important, yes. And
Well,
exactly.
The choir
Yes.
here.
We're privileged to have you, and I'm so grateful that a family member was able to witness this. That's a joyful thing. Thank you, everyone who's in this room, because honestly, we have a hero here. So thank you.
Me now?
Yeah.
Can you hear me now?
Yes, perfect. Excellent.
Oh, by the way, I'm a Bronx Science graduate also.
What class?
A long time ago, long before you. Class of, I think next year is my 75th anniversary.
Yeah, I'm only on 55, so that is a while before.
And yeah. Now, the thing I find basically with the government is they have an enormous database somewhere. Okay. In fact, in my experience with the Department of Defense particularly, is that they always collected the raw data. To the point, for example, in the Iraq War, we didn't have the networking capability to handle it all. And all the data that was taken in Iraq, for example, was ultimately sent back to Fort Belvoir, I guess it was at that time.
Mm.
And I found the same thing true when I was working with some of the pharmaceutical companies. That was also a while ago, where the government, NIH, wanted all the raw data. Okay.
It's still a problem for every company I know of, that the government, whether it's government or publications or the like, there have to be archives suitable for all the raw data. And working with FDA particularly, the
Sure.
task was, no matter what kind of analysis you ran, you had to be capable of rerunning it at some point if some lawyer somewhere challenged your results. So you would typically end up, I know many people work with containers with the idea that that they have the software permanently encased in the appropriate environment. We similarly had earlier techniques that very much involved, I need to be able to reproduce that analysis we did 20 years ago. Yeah.
Yeah, but the problem now with what's called artificial intelligence, which is hardly artificial, but is that there is an enormous database that hasn't even been explored yet, especially in the medical field. And the question is, how do you go about putting it in a usable form?
I think that absolutely the hardest part of using AI in medicine is the critical eye of what you allow it to train on. The problem with large language models is they train on everything on the internet, whether it be true, false, manufactured for evil, or whatever. Whereas in a medical environment, you absolutely need things curated. You need the equivalent of an army of librarians to allow AI to process, starting from what they have. So the success of something like AlphaFold is based on a very limited collection of existing literature and the ability to compute from that. That isn't always true of the rest of training AI.
Right. Yeah. Okay.
Thank you so much, everyone. I'm going to stop the recording now.